is like, what would you need to model of the full internals of a cell so that you could do experiments on the virtual cell, and what those experiments, in silico, and those predictions would be useful for you to save you a lot of time in the wet lab, right? That would be the dream. Maybe you could 100x speed up experiments by doing most of it in silico, the search in silico, and then you do the validation step in the wet lab. That's the dream. And so, but maybe now, finally, so I was trying to build these components, AlphaFold being one, that would allow you eventually to model the full interaction, a full simulation of a cell. And I'd probably start with a yeast cell, and partly that's what Paul Nurse studied, because the yeast cell is like a full organism that's a single cell, right? So first of all, pairwise, proteins with proteins, proteins with RNA and DNA. But then the next step after that would be modeling maybe a whole pathway, maybe like the TOR pathway that's involved in cancer or something like this. And then eventually you might be able to model, a whole cell.